welcome to today's session. I was just telling TikTok this about past paper questions. They are AQA themed. They're not literally AQA questions, but they're based on past paper questions from AQA, and it is for A-level biology. So that is what we're going to be going through in today's session. I've got the first question here already. It's 5 marks. I'm going to zoom in a little bit. And we'll get started. So hopefully you can see, feel free to join in. Thank you for all the support I can see over on TikTok. Some really nice comments coming in. I'm so pleased that my videos are helping you. I have actually just scripted, and I'm going to be filming, I filmed half of it today, half of it tomorrow, about 10 different posts that are common mistakes that came up in the exams last year. So I've spent a long time going through the questions, the mark scheme, the examiner's feedback to pick out common themes to share with you so that you don't make the common mistakes that they did last year. So hopefully you'll gain more marks. I've seen a few people asking, when am I going to start doing some more work on the essays? Because I do quite a lot of work on essays every year. That is going to be probably from March time. So that is when I tend to kick off all of the essay work. Right, without further ado then, let's jump into these exam questions. So this first question, it says proteins can be organised into primary, secondary, tertiary or quaternary structure, and we have to describe, so that's the command word, describe how a quaternary protein is formed from amino acids. So it's how we get a quaternary structure from amino acids, and this is a 5-mark question. So 5 marks for this one. So first thing then is we're starting with these amino acids. So I'm going to begin then with mark number 1, saying that those amino acids joined by peptide bonds. And also I'm going to say via condensation reaction. So that now gives us the primary structure. I'm then going to say that the secondary structure is held by hydrogen bonds. There's always marks for talking about the bonds, so that's why I've gone peptide bonds would be in the primary. In fact, amino acids joined by peptide bonds, I'm going to say to form primary structure. Secondary structure is held by hydrogen bonds, and you could add in there, you have alpha helix or beta pleated sheet. The tertiary structure is the further folding held by ionic disulfide and hydrogen bonds. I have actually seen in more recent exam papers that sometimes they insist that you say between R groups. So I'm going to add that in because that has come up more frequently. So that is the location of the bond. It's between the different R groups of different amino acids. So then lastly, that takes on to the quaternary structure. So our quaternary structure is more than one polypeptide chain. Okay, right, let's just pick out the key marking points then. So if you are having a go, or if you're not, then at least you can pick out the key marking points. You could always turn this into a question that you could have a go at. So we've said peptide bonds, condensation reaction, hydrogen bonds, tertiary structure, we've got the ionic and disulfide bonds, quaternary, more than one polypeptide chain. And we have got 5 bullet points, so that would be our 5 marks. Right, that is question 1. I'm just going to quickly check all my screens to make sure you can still see. And in case I've missed anything, no, that's looking all good. Let me just check my screens on my laptop. Instagram, you seem to have a pretty good view, brilliant. And then, let's see, YouTube, better mute myself. Yeah, cool, looking good. Let's move on then to the next question. This time it's 6 marks, so a 6 marker, and it's the nucleus in eukaryotic organisms contains genetic material. It's another describe, but this time it's describe the structure of this genetic material. So what I'd be thinking is they've said it's eukaryotic organisms. So I'm going to be doing a note to myself, DNA in chromosomes. Because it said the nucleus and eukaryotes, so we're describing the DNA and the chromosomes basically. So that's what we'd need to do. So I'm going to start then. It was 6 marks, so we do need quite a few points. So I'm going to start by describing DNA. So I'm going to say DNA is a, so they said describe the structure, so that's why I'm describing the structure of DNA. DNA is a polymer of nucleotides. Now, I don't need to talk about how those join together because it just wants the structure. However, I am going to say held by not hydrogen bonds, sorry, phosphodiester bonds between those nucleotides. So held by phosphodiester bonds. I'm actually going to talk about the nucleotide itself because that's also part of the structure. So nucleotide equals deoxyribose, phosphate, and nitrogenous base. Right, this is 6 marks, we're up to 3. Next, I'm going to say 2 DNA chains are held by hydrogen bonds. I'm going to say forming a double helix. Okay, then I need to start. So we've got 1, 2, 3, 4. So I need 2 more points, and I've not said anything about chromosomes yet. So then if we start thinking about the arrangement of the DNA in the chromosomes in eukaryotic organisms, I'm going to say that the DNA is associated with histones. And then I'm going to describe our structure of a chromosome. So I'm going to say chromosome consists of 2 chromatids held by a centromere. So that's 1 chromatid, and that's the other one, and that's the centromere. Right, let's pick out our key points. So we've said polymer of nucleotides, phosphodiester bonds. I then described the components, hydrogen bond to make that double helix, histones, and then 2 chromatids with the centromere. So that would be the key marking points for this long answer AQA style A-level exam question. Right, let's see what we've got coming up next. So for this one, genetic variation is introduced to populations of species through different mechanisms. One mechanism is mutations, but we have to describe and explain the other mechanisms that increases genetic variation within a species. So we're describing and explaining, and this is 4 marks. So we can't do mutations, that is already considered. So I'm going to jump in with random fertilization of gametes. So that's 1. Then I'm going to think about the 2 that happen in meiosis. So we have independent segregation of homologous chromosomes in meiosis. And we also have crossing over. So crossing over between homologous chromosomes. Oops. But at the moment I've just described, I haven't really explained. So what happens, particularly with these 2, is it introduces genetic variation because it produces new combinations of alleles in the gametes. And that's how we get the genetic variation. So the key concepts then: random fertilization, independent segregation, crossing over, and we've got new combinations of gametes as our explanation. Right, next on the list is a 5 marker. So for this 5 marker, we now have water is essential for organisms to survive. We have to state and explain 5 properties that make water essential for organisms' survival. So looking at these essential properties of water. So there's 5 that you need to know for AQA. We have metabolite, solvent, high heat capacity, large latent heat and vaporization, and then the concept of cohesion. So those will be the 5 marks, but it's not just stating it, you have to explain as well. So for number, oops, no, don't want highlighter. For number 1, I'm going to say metabolites. And then the explanation, they accept you just basically stating some examples of chemical reactions. So I'm going to say in hydrolysis, for example. But you could say in a condensation reaction, photosynthesis, respiration, but I'm just going to stick to that one. Number 2, I'm going to say solvent. And the reason that's useful is there's 2 different things you could say. So reactions can occur. Or you could say, so it allows the transport of substances, because they'll be transported, dissolved in maybe water in the plasma of the blood or water in the xylem. So it allows reactions to occur, or it allows the transport of substances. Either of those would be that second marking point. Number 3, I'm going to go for high heat capacity. You don't actually have to say specific heat capacity to get the mark. So high heat capacity. And that one, the mark is always, the advantage is it buffers temperature changes. Number 4, large latent heat of vaporization. The reason that's important is it provides a cooling effect. And then the last one we said was cohesion. And for this one, you could say that provides surface tension, so it'll support like a habitat for small organisms. Or you could have cohesion, so it supports a water column, in the idea of like the transpiration stream. So that's what I'm going to go for. One thing I just want to point out as well is this. High heat capacity, large latent heat of vaporization. You cannot get those mixed up. So you can't say high latent heat. It has to be large latent heat, and it can't be large heat capacity. So the reason that I've underlined those is to try and help you remember it should be the two H's together and the two L's together. High heat capacity, large latent heat of vaporization. So you do have to have it that way around. Okay, let's see this next one. I think this might be the last one today. So we've got DNA replicates through semi-conservative replication, and we have to describe the process of semi-conservative replication. And this one is 5 marks. So let's start with, we have to think about the two enzymes involved. There's definitely going to be marks for those. So that's DNA helicase and DNA polymerase. DNA helicase is used first, so let's start there. DNA helicase, and that is going to break hydrogen bonds. And that breaks it between those two strands of DNA, so they then separate. And once separated, both DNA strands act as a template. Then the third step is free-floating nucleotides are going to align opposite their complementary base pairs. Now those have lined up, this is where we then get the second enzyme, DNA polymerase. And as the name suggests, wherever you see ASE, that means it's an enzyme. And if this is DNA polymer enzyme, that's what ASE means, that's what this enzyme does. It creates a DNA polymers. So DNA polymerase joins nucleotides, and that creates the new strand. And I'm going to say forming phosphodiester bonds, because it's always good to name the bonds. It's not hydrogen bonds. Hydrogen bonds are between or opposite complementary bases. To get your new DNA chain, that would be phosphodiester bonds, but between, that's where you have the hydrogen. But these bonds between the nucleotides is phosphodiester bonds. Right, we've got 5 marks. Although I'm going to add an extra one because it says semi-conservative. So I'm going to talk about the fact that semi-conservative means each new DNA molecule consists of one original. B, what are you doing? My cat's climbing the wall again. Do you remember she did this last week? She's literally trying to climb the wall. I think she thinks, oh, she's distracted, I can get up to no good. Each new DNA molecule consists of one original strand and one new strand. Right, let's pick out some key points. So always talk about the enzymes. DNA helicase, breaking the hydrogen bonds. Both are acting as a template. The idea of the complementary base pairs, DNA polymerase joining nucleotides, phosphodiester bonds, and then we had our definition, one original strand, one old strand. So yeah, that is it. We've got through all of those long answer questions for today. I hope you found that helpful, having a go at some of those long answer questions with me, and just seeing the key marking points and just picking out some particular things. Oh, I was going to show you B because I was like, she clearly wants attention, but she's run off now. So we'll end it there. And if you've got any requests for next week, let me know. But that is it for today, everyone.